Hello, good afternoon. Thank you for the invitation. I apologize that it has to be pre-recorded because it would actually be midnight my time if I were to give this talk. So let me first start by putting up the presentation. All right, let's see. Okay, today I'm going to talk to you about a story that was published in 2018 on cortical interneuron migration. And this story is really the basis of the data from this work actually guide us to look carefully on shared and unique developmental programs that are important for the maturation of cortical inhibitory neurons. And if you have any questions throughout the talk, please feel free to send me an email. I'll try to answer it as soon as I can. So my lab is interested in understanding how the brain is built. The region of the brain we work on is the mammalian neocortex, which houses some of the most complex circuitry. And the cell type that my lab study extensively are inhibitory neurons, which, unlike the main types of neurons in the cortex, which are excitatory and that they project long distances, these neurons are mainly locally projecting and inhibitory. I say mainly because I'll come back to this point later. Now, they are very important because they control local circuit. And I give you an example. For example, if you're at a noisy party, you can still have a relevant conversation with someone because your brain is able to gate out all the excessive irrelevant information and let you carry out this conversation. This gating mechanism is also quite important because it prevents the cortex from going into overdrive due to excessive excitation. And as such, defects in the development of these cell type has often been associated with the cause of major developmental disorders such as epilepsy, autism, and attention deficit disorders. So now, this diversity that I talked to you about allows our brain to gate information very accurately, and it's very important for normal brain function. One very important fundamental question that we've been very interested in answering is how this inhibitory neuron diversity is generated during development. Because what you see here depicted in this cartoon is that they're diverse not just by morphology, they're diverse by biochemical class, the markers they express, as well as how they form synapse in different subcellular location also is integrated in their diversity. But yet, if we go back in time and we look in development at the progenitor level and newly born neurons, all of them more or less look the same. So what are the key events that happen during development that allows for neurons that more or less look the same to each other to having this highly diverse structure and function? To get at this problem, we started the project looking at one key event, which is migration. We know that cortical inhibitory neurons are born outside the cortex in the ganglionic eminences, abbreviated here as medial ganglionic eminences, MGE. So the bulk of the inhibitory neuron comes from this region in the ganglionic eminences. And to get to the cortex, they perform a long tangential migration via two very distinct routes, one at the marginal zone and the other one at the subventricular zone. So it's been long hypothesized that this dual route give special microenvironment that allows for this diversity, at least as one of the mechanisms. So we started the project asking a very simple question. Do different types of interneuron take different route to get to the cortex? Even though interneuron diversity, as I depicted to you, is large, and more recently, single-cell sequencing experiments have been able to give markers to each of these subpopulations. However, this is done in the adult. During development, we cannot discern every single class or subtype of these populations. What we can mark are full populations that come from the caudal ganglion eminences, full population that comes from the medial ganglion eminences, as well as subpopulation, the somatostatin expressing interneuron. So with these transgenic Cree-line, we started to assess the migration pattern of different major groups of cortical inhibitory neurons. So here I show you a comparison between the entire MG population using the NKX 2.1 Cree-line cross-reporter, as well as a subpopulation within this, the somatostatin population. And what you can appreciate just by looking at these images is that there are relatively a lot more cells taking the marginal zone route in the somatostatin Cree population. We have also used a different line that marks a subset of somatostatin cell. And I'll come back to why this line is important. But no matter what developmental stage we looked at, the fraction of interneuron passing through the marginal zone is more or less 20 to 30% at most of all developmental stages we examine in both the MG-derived population as well as the CG-derived population. However, in the somatostatin population only, what you see is there's a huge enrichment of cell passing through the marginal zone. So these data tells us different subtypes of interneuron, even with this kind of crude marking of cell population, migrates with different preference in different routes. And because of this, we now ask the question whether or not the population here at the marginal zone completely differ in identity to this of the subventricular zone. To do so, we devised a way to physically separate the two migratory streams by generating a horizontal type sectioning, a bit like a whole mount, and cutting from 100 micron from the top, allowing us to separate marginal zone migrating cells with subventricular zone migrating cells. And we can perform, then what we did was to perform FACS experiment to purify only somatostatin cell and compare the gene expression pattern. What we find is that there are about 80 genes that are differentially expressed among marginal zone versus subventricular zone migrating somatostatin cell. This is the first level of analysis where we find all the genes that are differentially expressed. What we really want to know is whether or not the cells that are taking the marginal zone route is indeed a different subtype than those taking the subventricular zone route. So we're a bit lucky because at that time, the Allen Brain Institute had just published single cell sequencing data in the adult neocortex and found that within the somatostatin population, there are at least 6 subtypes of somatostatin cell by transcriptomics approach. And so we were able to download these data set and ask the genes that enrich in marginal zone, where among these 6 different populations are they also enriched in? And when we do this analysis, what we find is that these marginal zone genes, these genes that are enriched in the marginal zone, are also highly enriched in 3 populations, the CDK6, cerebellum 4, and MYH8 population, telling us that marginal zone migrating cells share similar molecular signature with these 3 populations of interneuron. We can do the same experiment with the genes that are enriched in the subventricular zone. And what we find is that subventricular zone migrating cells appears to be also enriched, subventricular migrating genes and the cells shares very similar molecular signature as the shoulder positive population. And I'll come back to this a little bit later, but let me move on. And so we focused the paper on these marginal zone enriched population, because for the following reason. We know that by morphology and by electrophysiology approach, somatostatin cells can be divided into Martinoti and non-Martinoti based on their axonal arborization. As you can see, a Martinoti cell has large axonal arborization in layer 1. In contrast, the non-Martinoti cell are locally within their laminar layer projection, or basically they don't project to layer 1. And this is also seen in somatostatin cells in layer 2-3, you really have two groups. You have Martinotti as well as the non-Martinotti. And how this link, we know that in the Allen Brain single-cell transcriptomic paper, they were able to link one of their populations, transcriptomic population, the cerebellum 4 population, as layer 2-3 Martinotti cell. So we know that this is the population that share similar molecular features with cells that are taking the marginal zone. So the model we came up with is as follows. We think that Martinotti cells take the marginal zone to get to the cortex. This means that if we are able to label a migrating cell at the marginal zone, one would expect that by the time they become an adult, they'll all be Martinotti cells. Okay, so that's the idea. And in order to test this hypothesis, we had to do a little bit of work, which I'm going to explain to you, take you through quickly some of these data. So the first thing we need to do is we need to be able to label and estimate the population of Martinotti cells in layer 2-3. Because if they're all Martinotti cells, then obviously, no matter what root we label, we're going to label Martinotti cells. We know from literature that the distribution appears to be 66% Martinotti cells in layer 2-3, and the rest are non-Martinotti cells. So to verify this, we're able to inject a low titer retrovirus that express tomato in a conditional manner in the somatostatin-free animal at E14.5. Using this method, we know that we will mainly label layer 2-3 somatostatin cell. And because this method is very sparse, it allowed us to look at the full morphology, and we can quantify how many cells we label are somatostatin and how many cells we label they are are Martinotti, and how many cells we label are basically Martinotti or non-Martinotti. And the proportion comes up to be about 60-40, matching what was in the literature. Okay, now this type of labeling is labeling progenitor, so it's unbiased, and it randomly labels cells from either root. Now, the second approach we take was we need to find a way to label cells that is passing through the marginal zone in vivo and ask what they become when they get to adult stages. So the method we devised was a pure electroporation method where we inject DNA right underneath the skull and electroporate. In this, I'll show you first the control for this. We took a LHX6 Cree animal, which labels MG-derived interneuron, and we electroporate both Cree-dependent YFP as well as a non-Cree-dependent M-cherry. What you could see is that we label a lot of cells in the M-cherry, a lot more than the Cree-dependent one, which is logical. But all the cells we label are sitting in the marginal zone. And so this method tells us that we can label cells accurately in the marginal zone. And because of the sparseness of this labeling, by adult stages, we can also reconstruct morphology. So using this pure electroporation method, we now have a way to look at the somatostatin cells that are passing through the marginal zone. So here is the experiment where I show you already the control, where we can label both roots with the retrovirus. And when we do label it with the pure electroporation, what we find is that we enrich significantly for Martinotti cell. What this tells us is that that the cells taking the marginal zone eventually becomes Martinotti cell. So from this data, we came up with a model of why migrating through the marginal zone would be critical for these particular type of neurons. And the model is largely inspired by granular cells in cerebellum. As some of you might know, granular cells also transition from tangential migration to radial migration. And when they do radial migration, they usually leave behind the trailing neurites that later become the parallel fibers. We believe the same thing is happening in the cortex for these Martinotti cells, that they would take the marginal zone, turn into the cortical plate, leave behind trailing neurites that later become the nascent axon and the full. And as they mature, the nascent axon becomes a full axonal arborist, as you've seen in an adult Martinotti cell. this means that the implication for this is that migration at the marginal zone is therefore a key developmental process for the maturation of these type of cell. Now, what I've shown you is that indeed a cell that migrates at the marginal zone eventually becomes a Martinez cell with this peer electroporation method. What I have not shown you is whether this part of the model indeed is true. So to do so, we turn to culturing slices at postnatal day 0, cortical slices, to maintain the 3D morphology where we have the marginal zone, surface of the cortex on top, and the cortical plate here in the bottom of the image. So I first show you a control in a line that we know mainly marks non-Martinez cell. And we are tracking both a cell soma at the marginal zone in blue circle, as well as their trilling neuroid tips in the marginal zone in red. And as I play this movie, what you would see is that in the control condition, as an interneuron enters into the cortical plate, its trilling neuroid is highly dynamic and almost goes with it. In contrast, the same type of movie in a line that mainly marks Martinez cell, what we see is quite different. The cell migrates into the cortical plate. However, its trilling neuroid almost doesn't move out of the marginal zone. It's really being left behind as the cell moves down. So it's growing an axon as it's moving down. And we can do this quantification by looking at the speed of cell soma entering the cortical plate, migrating in the cortical plate, as well as how fast the neuroid tip is moving. And as what you can see here, the neuroid tip in the Martinez cell line is more or less not moving at all. So collectively, what this data tells us is that migration and axonal targeting are directly coupled to optimize their circuit formation. And in the paper, which is what I'm not going to talk about, we also have a genetic model that we can perturb this migration, and we showed that indeed the axonal projections and the arborization is completely altered due to this mutation. So, but more importantly, what this tells us is also that we tend to think of developmental programs as sequential, where you have neurogenesis first, and then migration and axon projection, and then synapse formation. But in fact, the framework to make a synapse or to target an axon, in this case, starts at migration. And it's thought to be long before, probably weeks before the first synapse occur. The second thing these data set tells us about is that when we look at maturation as a population, we actually miss a lot of information, because we know from the textbook, this kind of scheme is what you see classically in the textbook, where a neuron is born, it migrates, it goes to its laminar location. There's a programmed cell death around first to second postnatal week, and then axon and synapse refine itself. Okay, what I've shown you in this work is that at least in the process of migration, we have unique developmental trajectory. For example, the Martinez cell takes the marginal zone and leaves behind these trilling neuroids to get into the cortical plate as part of their axonal targeting processes. Revealed in this data is there's another group of cells that are these long-range somatostatin cells. Because of their marker with total, we know that these are indeed long-range somatostatin cells. So they're not interneuron anymore. They're actually projecting cells that are inhibitory from the cortex, and they take the subventricular zone. We now have a platform to look at the unique maturation of these long-range cells, because we've performed sequencing experiment and we know what genes are enriched in this population versus these population. And one of these genes is a transcription factor called POOF3F2 or BRIM2. We follow up our observation with the migration data with single cell sequencing data, where we fax out somatostatin cell at P1. As you can see, they're all GABAergic, and we cluster them using unsupervised clustering. What we find very easily at P1 is that one population of cell sits almost uniquely different than other population, and these cells ends up being long-range cell as shown by the marker chodel. These are also the cells that express high levels of PUF3F BRIM2. So we found a gene that was enriched in the subventricular zone. Now we confirm that it's uniquely expressed in long-range somatostatin cell in the adult by the data from Allen brain. We know that these are also uniquely in long-range cell. So now we have a gene that is expressed in a very acute manner, in a very acute cell type, and we ask, maybe this is also this marker is also something that would mark them earlier in development when they're in progenitor state. So before I go into that, let me describe a little bit on what these long-range cells are first. We know that these have been characterized for almost a decade, but there's actually surprisingly little that's known about them. Her et al. At 2016 used NOS and somatostatin as a marker to mark them and generated various combination transgenic line. And they show that these can be cortical projecting, projecting from either the visual cortex to the motor cortex or vice versa. Metzler et al. Have shown that if you inject anterogradely an AAV virus into the motor cortex, you would see projections in the striatum. So they can also be cortical striata. More recently, the projection patterns that is publicly available on the Allen Brain Institute website showed that an injection in the barrel cortex will also result in projections in the striatum in the somatostatin Cree mouse line. So with these, we start to ask, how early are these projection patterns established? What are the molecular mechanisms that drive the maturation of this cell? And is there a progenitor zone that is biased in generating these cell types? So in the interest of time, in the next 3-4 minutes, what I'm going to go into is talk more or less about point 2 and point 3. So the molecular mechanism we think are driving the maturation of these cell types, one of the candidates would be PUF3F2 BRIM2 because of their acute expression in these different cell types. Now, we also know that when we do a staining and immunostaining, we find PUF expressing in different progenitor types in the MGE. To characterize this a bit better, we collaborated with Resolve Bioscience, which has a pre-commercial release platform of spatial transcriptomics based upon the Murfish method. So what this does is, in the end, it's really a single molecule fish experiment that you can perform up to over 100 to 200 transcript. So we started a pilot with them, and we examined about 25 transcript on E12.5 tissue in the MGE. So this is a bit an overview of an experiment on how it's done. Each color that you see here represents a different probe. We also developed a method for us to segment cells in the progenitor zone, which are highly packed with each other with a membrane marker staining. And so right now, we can accurately detect where the cells are, as well as up to 100 transcript in this method. Now, I'll show you some preliminary data with just a trial set of genes that marks different population of progenitor. We use these are mainly transcription factor. And what you can see very clearly is that in the dorsal MGE, we can actually identify 3 different types of progenitor. One in the dorsal region, one in the ventral region, and 2 in the dorsal region, in fact, that express high levels of PUF, one of them with also co-expression with the hip. So we posit that PUF is not only important for maintaining a maturation of these long-range somatostatin cell, but it's also important to drive the fate. So basically, in summary, what I've shown you is that root-enriched gene has given us an entry point to examine cell type-specific maturation, and PUF is one of our candidates that appears to be highly enriched in these long-range cell. And we have used spatial transcriptomic approach to reveal the progenitors that could be critical for the generation of these different cell type. So in the future, obviously, I hope that we'll be tell you the story on what happens when we remove proof from somatostatin cell from the progenitors. Finally, I end my talk here with acknowledging, I've tried to acknowledge most of our collaborators and students who have done the work. We are a new lab in Belgium, and we are actively recruiting postdoc students. So please feel free to drop me a line. Thank you very much.